Hi everyone, this is Ram. Today we are going to discuss about
Biological Safety Cabinets, BSCs. Slide 2. Introduction to Biological Safety Cabinets, BSCs. BSCs provide protection by safeguarding users, experimental samples, and the environment from biohazard exposure. They are essential in labs handling infectious agents, GMOs, and pathogenic microorganisms. 3 levels of protection: O personnel, HEPA filters, and an air curtain shield user. O sample protection. Recirculating and unidirectional HEPA filtered air prevents contamination. O lab environmental protection. HEPA filtered exhaust safeguards the lab environment. Key distinction. Unlike laminar flow hoods, BSCs protect the user and the environment, not just the product. Slide 3. This diagram shows the airflow and filtration system inside what appears to be a biological safety cabinet, BSC, or a similar clean air containment device. Here's the explanation of the parts and flow. 1. Polluted air. Air inside the working area may contain contaminants. 2. Outside air. Fresh air is drawn from the surroundings into the cabinet. 3. Pre-filter. This first filter removes large dust particles and debris from incoming air before it enters the main system. 4. Fan pulls in the outside air and recirculates part of the internal air. 5. Mixed airflow. Combination of recirculated clean air and incoming outside air is moved through the system. 6. Recirculation air filter, usually a HEPA filter that removes fine particles and microorganisms before air is sent into the working area. 7. Clean air filtered. Particle-free air flows down into the work zone to protect the product from contamination. 8. Exhaust air filter, another HEPA filter that cleans contaminated air before releasing it outside. 9. Exhaust clean air that has been fully filtered and safely released into the room or ducted outdoors. How it works in sequence. Outside, air enters through the pre-filter. The fan draws this air in and mixes it with recirculated internal air. The recirculation air filter purifies the mixed air before it enters the work zone. Inside the work zone, clean air flows downward, protecting the sample. Contaminated air from the work zone is pulled into exhaust channels, filtered through the exhaust air filter, and then released as clean exhaust. Slide 4. The principle of operation. Airflow patterns. Room air enters the front grille, and contaminated air is filtered before being exhausted. Filtration. HEPA filters trap 99.97% of particles greater or equal to 0.3 microns. ULPA filters offer even higher efficiency. Containment zones. Negative pressure within the cabinet prevents contaminated air from escaping. Common component. All BSC classes share a high-efficiency particulate air HEPA filter. Slide 5. Types of BSCs. An overview. Class I. Provides operator and environmental protection, but no product protection. Class II provides protection for the operator, product, and environment, widely used in hospitals and research labs. Class III. A gas-tight glove box for maximum containment of BSL-4 pathogens. All 3 classes use HEPA filters, which eliminate 0.3 micron-sized particles, including bacteria, spores, and viruses, with 99.97% effectiveness. Slide 6. Class I. Biological Safety Cabinets. Most basic type of safety cabinet. Provides personnel and environmental protection only. Operates with an open front and negative pressure ventilation. Incoming air is not filtered, so it's not suitable for materials that must be handled under sterile conditions. Working mechanism. Air enters the front, moves over the workspace, and exits through a HEPA filter, trapping pollutants. Slide 7. This diagram illustrates the working principle of a Class I biological safety cabinet, BSC. The arrows show the direction of airflow, highlighting how the cabinet protects the user and the environment, but not the samples inside. Airflow explained. Inflow, blue arrow. Room air is pulled in through the front opening of the cabinet. This creates a barrier of inward airflow that protects the user from any contaminants inside the cabinet. upward flow, orange arrow. An internal fan draws the air from the workspace upward. This air contains any aerosols or contaminants generated during the work process. Filtration and exhaust, blue arrow. The contaminated air is forced through a HEPA filter, which traps a large percentage of airborne particles like bacteria and viruses. After filtration, the clean air is exhausted from the cabinet, ensuring the surrounding environment is not contaminated. In short, a Class I BSC works by containing and filtering contaminants before they can reach the user or the room. It does not protect the materials on the workspace from contamination because the incoming air is not filtered. Slide 8. Class II Biological Safety Cabinets protects people, the environment, and the product. Commonly found in clinical, life science, and pharmaceutical labs. Filters the air supply to the work area using laminar airflow to reduce turbulence. Types, further distinguished by construction, ventilation, and exhaust systems. The main types are A1, A2, B1, B2, and C1. Key features: O downward HEPA filtered unidirectional airflow for product protection. O open front with inward airflow for personnel protection. O HEPA filtered exhaust air for environmental protection. Slide 9. Class II subtypes. A1 and A2 type A1. 70% of air is recirculated and 30% is exhausted. O inapplicable for use with toxic chemicals and radioactive nucleotides. Type A2. O also recirculates 70% of air and exhausts 30%. O permits the use of radionucleotides and toxic chemicals if connected to an external exhaust system. This diagram illustrates the airflow in a Class II Type A2 Biological Safety Cabinet, BSC, which provides protection for the user, the product, and the environment. Airflow explained. Inflow, green arrows. Room air is drawn in through the front opening of the cabinet, creating an air barrier that protects the user from any contaminants inside. Recirculation, red arrows. An internal blower draws the air from the workspace into a common plenum. Approximately 70% of this contaminated air is then pushed through a HEPA filter and recirculated back into the work area as clean, downward-flowing air, light blue arrows. This provides a sterile environment for the product. Exhaust, dark blue arrows. The remaining 30% of the contaminated air is directed to a separate plenum, passes through a second HEPA filter, and is then exhausted back into the room. In essence, the Class II Type A2 BSC uses both an inflow air barrier and a downward curtain of sterile air to protect the user and the product, while also filtering and exhausting some of the air to protect the environment. Slide 10. This diagram compares the airflow patterns of two types of biological safety cabinets. A Class II Type A2 on the left and a Class II Type B2 on the right. Both cabinets provide protection for the user, the product, and the environment. Class II Type A2 left. This is a partially recirculating cabinet. Airflow, room air, blue arrows, enters through the front grille. The internal fan, labeled B, pulls contaminated air, red arrows, from the workspace A up to the top. Filtration and exhaust. This air is then split. A portion of the air, typically 30%, is pushed through a HEPA ULPA filter and exhausted out of the top of the cabinet, green arrows. The remaining air, typically 70%, is pushed through a second HEPA filter and recirculated back into the work area, A as clean, sterile air. Purpose: this design balances containment with energy efficiency, making it the most common type of BSC. Class II Type B2, right. This is a total exhaust or non-recirculating cabinet. Airflow. Room air, blue arrows, enters through the front grille. All of the contaminated air, red arrows, from the work chamber A is directed to the top of the cabinet. Filtration and exhaust. The contaminated air is then pushed through a HEPA ULPA filter and 100% of it is exhausted from the cabinet, green arrows. No air is recirculated. Purpose. The complete exhaust system makes this type of cabinet suitable for work involving volatile toxic chemicals or radionuclides, as there is no risk of recirculating hazardous fumes back into the workspace. Slide 11. Class II subtypes, B1 and B2. Type B1, O, a hybrid of Type A2 and Type B2. O uses both an internal blower, recirculating 30% of air, and an external blower, exhausting 70%. O ideal for work with small amounts of volatile toxic chemicals. Slide 12. This diagram shows a Class II Type B1 Biological Safety Cabinet, BSC, a type of ducted cabinet that offers protection for personnel, the product, and the environment. The airflow is split into two paths, a recirculated portion and an exhausted portion. Inflow A, room air enters through the front opening A. Recirculation, an internal blower F pulls a portion of this air, along with some from the workspace, through the supply HEPA filter D. This filtered, clean air then flows downward over the workspace, protecting the product. A portion of this air is then recirculated back into the system. The note about additional HEPA filter for supply air G indicates a filter on the recirculated path to ensure cleanliness before it re-enters the supply plenum. The dotted lines show the negative pressure areas. Exhaust. A dedicated external blower pulls air from the negative pressure exhaust plenum, E. This air is routed through an exhaust HEPA filter, C, and is then completely exhausted out of the building. This is where the majority of the air, around 70%, is removed, making this cabinet suitable for working with small amounts of volatile chemicals. The sash B acts as a barrier, and the negative pressure in the plenum, E, ensures that contaminated air cannot escape into the room. Slide 13. Type B2, O, 100% air exhausted through HEPA filters. No air is recirculated. O requires a dedicated external exhaust blower to operate. O best for operations that need the emission of chemical vapors. This diagram shows the airflow in a Class II Type B2 Biological Safety Cabinet, BSC, a non-recirculating model that provides the highest level of protection among Class II cabinets. Airflow explained. Inflow A. Room air is drawn in through the front opening A, creating a protective barrier for the user. Contaminated airflow. An internal blower pulls this air and air from the work area upward and through a supply HEPA filter, D. This filtered air flows downward over the workspace, protecting the product. Total exhaust. All of the air from the work area is directed into a negative pressure exhaust plenum, E. From there, it's pulled through an exhaust HEPA filter, C, and is 100% exhausted out of the cabinet and into the building's exhaust system. No air is recirculated back into the work area. This total exhaust design makes the Type B2 BSC ideal for working with volatile toxic chemicals and radionuclides because there is no risk of recirculating hazardous fumes. Slide 14. Class II Type C1 and Class III Type C1. O can operate in recirculating mode, like Type A, or exhausting mode, like Type B. O features a designated exhaust work area for use with radionuclides or hazardous vapors. This diagram illustrates the airflow in a Class II Type A1 Biological Safety Cabinet, BSC, a partially recirculating cabinet. It's designed to protect the user, the product, and the environment. Airflow explained. Inflow A. Room air is drawn into the cabinet through the front opening, creating an air barrier that protects the user. Recirculation. The air inside the cabinet, now containing contaminants, is pulled up by the supply blower, E. Approximately 70% of this air is pushed through a supply filter, D, and flows downward over the workspace as clean, sterile air. Exhaust. The remaining air, about 30%, is channeled by the exhaust blower, F, through an exhaust HEPA filter, C, and then released back into the room. The key feature of this model is the recirculation of the majority of the air, which makes it less suitable for work with volatile chemicals since some fumes would be recirculated. Slide 15. This diagram illustrates a Class II Type A2 Biological Safety Cabinet, BSC, that is connected to a building's external exhaust system via a canopy. This setup allows for the safe handling of small amounts of volatile chemicals by exhausting the fumes away from the lab. Airflow explained. Inflow A. Room air enters the cabinet through the front opening, creating a protective barrier for the user. Recirculation. An internal supply blower draws contaminated air from the work area and pushes about 70% of it through a supply HEPA filter D. This filtered, clean air then flows downward over the workspace, protecting the product. Exhaust. The remaining 30% of the contaminated air is drawn by the exhaust blower F through an exhaust HEPA filter C. This filtered air is then exhausted out of the cabinet's top into a canopy opening I, which is connected to a building's main exhaust system via a sealed flexible duct H and exhaust duct J. The canopy design creates a physical break to prevent a potential pressure imbalance from the building's exhaust system from affecting the BSC's internal airflow. Slide 16, Class III. O. Highest level of protection for people, products, and the environment. O. A completely sealed, non-opening enclosure with a dedicated exhaust system. O. Uses heavy-duty rubber gloves for work and is designed for BSL-4 pathogens. Slide 17. This diagram shows the working principle of a Class III Biological Safety Cabinet, BSC, also known as a glove box. It provides the highest level of containment and protection for personnel, the product, and the environment. Airflow and features complete isolation. The cabinet is a gas-tight, sealed enclosure with a negative pressure workspace. This prevents any contaminated air from escaping. Inflow. Air is drawn from the room into the cabinet through an inlet HEPA filter. This ensures that only clean, sterile air enters the workspace, protecting the product. Glove ports. The user interacts with the materials inside the cabinet using built-in, sealed glove ports. This maintains a physical barrier, ensuring the user's safety. Exhaust. All air is exhausted through 2 HEPA filters in series. This double filtration system provides a high level of confidence that no airborne contaminants are released into the environment. The exhausted air is sent to a dedicated exhaust system, ensuring it's safely removed from the building. Slide 18. Operational and safety guidelines before use. O. Switch on the blower, check for blockages. O. Allow the air to purge the workspace for 5 to 10 minutes. O. Disinfect all interior surfaces during use. O. Wear appropriate PPE, lab coat, gloves, mask. O. Avoid blocking grills. O. Minimize rapid movements to maintain proper airflow. After use, O. Allow a post-purge for the air to clear. O. Disinfect all interior surfaces again. O. Shut down the fan and lights. Slide 19. Applications and limitations. Applications. O. Used in diagnostic labs, pharmaceutical manufacturing, and public health monitoring. O. Suitable for procedures that create aerosols, such as vortex mixing, sonication, and centrifugation. Limitations. O. High cost and require professional operation and maintenance. O. Contamination can be caused by airflow disruption. O. Chemical vapors require specialized hoods as HEPA filters do not trap gases. Slide 20. Final summary. BSCs are foundational for laboratory biosafety, providing essential protection for personnel, samples, and the environment. Regular maintenance, annual certification, and adherence to safety protocols are crucial to ensure their effectiveness.